Hello, everyone. Welcome to today's live broadcast, Application of Targeted Next-Generation Sequencing Panels in Neurodevelopmental Disorder Research. I am Kristen Perret-Gentile of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. For more information, please visit labroots.com. This event is sponsored by Thermo Fisher Scientific Ion Torrent. Next-generation sequencing utilizes massively parallel sequencing to generate thousands of megabases of sequence information per day, opening doors to new research studies that were once difficult to accomplish in a practical manner. Powered by semiconductor chips, Ion Torrent sequencing technology is simpler, faster, and more cost-effective and scalable than other benchtop next-generation sequencing technologies. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can, and any questions we do not address today will be answered via email. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. And if you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button in the lower left. This is an educational webinar and thus offers free continuing education credit. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your web page and follow the process of obtaining your credit. I would now like to introduce today's speakers, Alessandro Murgia and Francisco Hernandez-Guzman. Dr. Alessandro Murgia is the Director of Laboratory of Molecular Genetics of Neurodevelopment Department and Associate Professor of Pediatrics at Department of Neurosciences and Department of Women's and Child's Health at University of Padua. Dr. Murgia's areas of interest include neurodevelopmental disorders, intellectual disability and autism spectrum disorders, early onset epilepsy, and hereditary deafness. Francisco Hernandez-Guzman is a Senior Product Manager for the Clinical Sequencing Division at Thermo Fisher Scientific. He manages the Ion AmpliSeq custom product line, including the automated custom design creation process in Ion AmpliSeq Designer and custom design services. Francisco has been working in the life sciences field for 20 years and has been a product manager for the past 7 years. And I will now turn it over to Dr. Murgia for her presentation.
Hello, everyone. So we will talk today about the application of targeted next-generation sequencing panels in neurodevelopmental disorder research. As I was presented, I would just go ahead and say that we are working at the University Children's Hospital in Padua. We are clinically part of the neuroscience section of our department, and the laboratory is set in a different building, as you can see it here. The Laboratory of Molecular Genetics and Neurodevelopment is in the Pediatric Research Building. This is our setting, and I guess what I want to say is we are pediatricians involved in neurodevelopment. We are, our scientific interest is focused on this, so we study genetic basis of conditions involving intellectual disability, autism spectrum disorder, early epilepsy of various types, sensory neurohearing loss. We, in the course of the years, have developed and are currently working with 4 NGS panels. 2 new ones addressing cortical brain malformations and cerebral palsy are in progress. What are neurodevelopmental disorders? This is the definition of a group of conditions that are clinically and etiologically highly heterogeneous and are characterized by a wide range of overlapping clinical features. Common features may include intellectual disability, autism spectrum disorder, speech difficulties, neurodevelopment retardations in general, seizure, microcephaly, ataxia, impaired sleep, and neuroradiological correlates. All these things are common or may be common to different conditions. So the diagnosis based on exclusive clinical criteria may be very difficult or just simply not possible. And on the other hand, etiological information are highly needed to provide insights into the pathogenic mechanisms of these conditions and help understanding the different phenotypes, and among the different phenotypes, the variable clinical expression, which is very high. So with this in mind, how many possible genes are involved? This is the question. How many disease genes should be investigated if we wanted to cover the etiologies of this condition? Up until very recently, or up until recently I should say, these questions, or the possibility of answering these questions, was hampered by technology, and they're now, and it's now possible to find the right answer with next generation sequencing technologies. But then the question is, so which technology are we choosing? Which are the possible NGS strategies, and how do we decide which strategy we want to use? So the two possible options, or the two main options, are whole exome sequencing and targeted gene panels. The first one, within the first one, is a primary option for cases with unusual phenotypes, phenotypes that would likely not be resolved by analysis of known disease genes, or in which it is not possible to hypothesize obvious etiologies. Phenotypes that have no good targeted panels that seem to give a reasonably high yield answer. On the other hand, targeted gene panels, we think this is definitely the strategy of choice in conditions in which a known cohort of disease or candidate genes can actually give a good detection rate, which is obviously, which is becoming obvious that is in particular a case for hearing loss. An important consideration to actually come up with the strategy is the analytical performance of exomes versus panels. So if we consider targeted gene panels, we can say that with this strategy, a much lower percentage of exon getting sufficient coverage. We want our sequences to be covered as best as we can. So the width of coverage and the depth of coverage. The depth of coverage may significantly impact on the ability to detect genetic alteration. On the other hand, if it's true that extent and depth of coverage are critical elements of a good analytical performance, then we have to also recognize that whole exomes are actually not really whole. And in fact, there are recent reports that have been published in 2013 and 2017 that indicates that in individual study for the identification of the etiology of rare Mendelian disorders, whole exome sequencing detected about 30%, had a detection rate at about 30%. So this is what we have to start with. So in our setting, we decided that opted for a targeted strategy. We chose the Ion Torrent NGS platform. Why did we make this choice? What is the reason? What are the reasons of our choice? Well, first of all, the Ion Torrent NGS, in our opinion, offers innovation within an innovative technology, so it was attractive for that reason. The PGM as a platform combines state-of-the-art NGS sequencing with features that respond to the needs of a single laboratory. And ours is not a particularly large laboratory. It's a very focused one. Our aim is to focus on individual genes and on specific gene regions. We wanted the possibility of obtaining a wider sequence coverage and a very high depth of coverage. We wanted a simple workflow, and we wanted to have a time to get a result that could be short, and we do have results in a matter of days. And another consideration is the throughput can be very high in a single sequencing run, which is in turn a more cost-effective system. These are our reasons, but so we find ourselves into targeted panels, and there's a big offer in the market. The market offers a lot. There are many companies offering panels of all sorts and kinds. So why would anyone to choose a customized panel? Why would you want to customize something that's already made? Well, for a series of reasons, I guess. Our aim at disease-focused panels, the fact that we have a very specific involvement in disease areas, and this is coupled with our capacity of a complete phenotypic characterizations in these disease areas for the tested cases. Our goal of studying the pathogenic role of disease-causing and, let's say, established candidate genes and their molecular pathogenesis. Then the need of sequencing at the high depth. I'm repeating it, but I think it's very important, not just because we are more confident that we detect variants with a high depth of coverage, but also because we may find ourselves in a condition of dealing with, for example, somatic mosaicism, which is a problem even in Mendelian disorders that actually is becoming more and more obvious. Then the need of including analysis of specific genomic regions of interest. Among these regions, some possibly residing outside the coding sequences, so that they wouldn't be obviously included. Then we made a choice of avoiding unsolicited results or incidental findings, so we want to choose what we're dealing with. And then the aim of generating translational, immediately actionable data. The process of developing, so what we think is the most appropriate tool is rather complicated, as it's a rather time-consuming, and it's a good job to do. So we think the starting point is a specific biological and clinical knowledge of the problem, and the possibility of a complete phenotypic characterization. This is the foundation of our work, on which then we add an extensive literature search, an extensive data mining process to get out all the possible information about the genes, about the conditions, about their relationships. And then gene prioritization, which is to say evaluating a wide array of different evidences, going from gene expression to function and networks to omic data with a great deal of manual curation. And I guess I'd say this is, if not the major, one of the most important steps in the process. Let me give you an idea of what we've done in the choice of our panel and what are the criteria we started with. Like, for example, our epileptic panel. We chose genes from what's available from the growing list of genes that are available in the literature. We chose 31 genes. I guess it's fair to say that this was a panel that we have developed in 3 years ago now, in 2014, with the AmpliSec Designer 3.6 version. We selected 31 genes associated with early onset epileptic encephalopathy or epilepsy with onset within the first year of life. And we found ourselves with a clinical clustering of genes, including Dravet syndrome genes, infantile spasms, epilepsy of infancy, different type of clinical pictures that would all be involved in, that would all be included in conditions within the first year of life. Among these genes, 15 genes were underlying autosomal dominant conditions, 9 genes autosomal recessive. There were some X-linked genes. It was a panel of 773 applicants with a coverage of about 98%. But the concept here is early onset. This is the selection. We know now that we have to expand this panel a great deal, but we've had very good result with this. Our hearing loss panel was chosen based on non-syndromic criteria. We wanted a panel for non-syndromic sensory neural hearing loss, and we chose this because this is the type of hearing loss that present more difficulties in the differential diagnosis. We came up with a final subset of 59 genes that represented 65% of known deafness genes, but about 90% of the deafness mutation that had been reported in non-syndromic sensory neural hearing loss, so that was very efficient. We selected genes strictly associated with non-syndromic hearing loss, genes frequently mutated in literature and public databases in Caucasian, which is the type of population we mainly have to deal with. We didn't include genes with only private mutation report or genes which function was not strictly ear-related. But in the clinical clustering, as I point out here, you can see we have 11 Usher syndrome genes. So why do we have a non-syndromic hearing loss panel with Usher syndrome genes? Because we have to deal with a young population mainly, we're pediatrician, and we have to know that there are conditions which onset is apparently, seemingly, non-syndromic hearing loss. So this is one of the reasons why we have to exactly know what we're doing, because if we didn't include these genes, even though these are syndromic, we would lose part of what is seemingly non-syndromic early on. So that was our point. A third panel, and I will end with this, is a comorbidity panel, yet another criteria. We wanted to intercept genes that were the cause of a comorbid clinical picture between intellectual disability and autism spectrum disorder. So we started out with 4,000 genes involved with autism spectrum disorder or intellectual disability, gathered from database and literature. Then we filtered by recurrence, clinical phenotype, gene functions, and pathway, and came up with about 1,000 genes, of which we then selected very strictly genes that were shared between the two areas and could be used for protein-protein interaction construction. We then added some direct interactor and or very frequent genes. And then with a great deal of manual curation, we made this, we finally got to the panel with the concept of the expected informative yield. We ended up with a final subset of 74 genes, which functional clustering included target of the Fragile X mental retardation protein, postsynaptic proteins, chromatin modifiers, and 8 hub gene interactors. And most of these were autosomal dominant genes responsible of autosomal dominant condition. 20 were involved with X-linked mental retardation. Some of them are autosomal recessive, but what I want to point out is that in this panel we did include all the known genes involved with the Rett Engelmann phenotype, which is a very important continuum in which the comorbidity between intellectual disability and autism spectrum disorder is a key feature. This is a larger amplicon, And again, this is our comorbidity panel, so it's a choice, it's a very specific choice. Now, but this is a lot of work. What I just represented is a lot of work. And so the question was, what if we had a large array of carefully prioritized genes to investigate specific clinical topics? What if we could access to something that was already studied in order to choose our gene? We then could choose from this sort of catalog and develop comprehensive multigenic panels, which is what we finally want to do. This is something that we had in mind, and we were fortunate enough that we had the opportunity of having an early access to this new ion AmpliSeq on-demand panel system. So we were very happy to try this new system and see how it worked. And we found ourselves browsing disease areas that with an intuitive and informative and friendly interface that gave us the opportunity of putting together a panel including genes in our areas of choice. I have to say that the disease gene catalog was somehow more limited than it is now and will become in the near future. So we came up with a 59 gene panel, which included about 1,800 amplicons with an amplicon dimension of about 200 base pairs, which we analyzed with a chemistry that's a new chemistry, the Ion AmpliSeq Library Kit Plus, and the Ion PGM High QView OT2 kit on 316 chips. The data analysis was initially done with the Ion Torrent Suite software 5.2.2 with a couple of plugins, the variant color and the coverage analysis, and the rest of the in silico analysis was done according to our current protocol. So the annotation, the processing and elaboration based on variant filtering, based on different criteria of filtering, so the coverage, the minimum allele frequency, based on our in-house database, based on the use of a combination of in silico prediction tools, and we then normally finish with Sanger validation and family segregation analysis, and this was done for this experiment as well. We have done 3 main experiments that were designed to study a new panel, which we didn't understand, which we didn't know before. So we based everything on our previous experience, and I won't go into details, but I am very happy to do it if there's interested about that. I guess the main concept here is we, in all the experiments, we had a very good result in terms of polyclonality, a very good result in terms of low quality, a very good result in terms of total reads we obtained, and a very good result in terms of clustering of read length, which was actually better than what we were used to having with our custom AmpliSeq designer and the previous chemistry. So very good result from this new system on top of the very good and friendly interface. So the question here was, okay, we had a few numbers here. We had on-target reads that were very good. The average reads per amplicon, the coverage was 176. The amplicon with no strand bias, very high. The end-to-end of the amplicons, and the same thing for the base coverage, which was very high with no strand bias and end-to-end reads. This is a profile of the coverage of our applicants that shows in all of our experiments the fact that we have a very low percentage of uncovered applicants or not enough cover applicants with a coverage less than 20%, which is the 20x, which is our cutoff. And these low coverage was predictably in regions with a high content of CG or regions that are known for being difficult to sequence, so we would expect lower coverage in this region. So we didn't have any surprise of low coverage in our panel. The overall average variance detection, the average variance per library was about 238, and the total variant we found was about 770, with about 200 private variants, of which very few with an insufficient coverage, and none of them was a frame shift deletion insertion that gives us the idea of a very low likelihood of errors here. So if it's only a few uncovered variants, we normally chase that too. We have a very good coverage of the private variants, generally speaking. And so these are, in our opinion, very good results and very consistent ones. Now we wonder, what are we looking at? Why are we getting this good result? Is this the on-demand panel, or is it the new chemistry that's involved in it? So if we look at our experiment and we want to figure out and we want to try and answer this question, we can compare this result to what we had before with our custom AmpliSeq and the AmpliSeq library kit and the AmpliSeq library kit that we used before. So what we did was we try another experiment with our custom AmpliSeq designer and with the new chemistry, and this is what we obtained. So obviously the AmpliSeq library kit, so the chemistry with which we now analyze, we now process our panel, is obviously very good and is obviously a key element in the good results we obtain. If you compare these two results, it's obvious that the chemistry make a definite difference. If we compare the result we have on the AmpliSeq Library Kit Plus and the AmpliSeq library that we used before on the same panel, we see that all of the important elements we want to consider, so the percent of on target, the end-to-end applicant reading, the end-to-end reads on base coverage, are all higher than what we used to obtain before, which was not a bad result, but this is a lot better. So I think we have answered the question, which one is the most important, or what is very important in these experiments. So in our view, how do we consider the system in general? We think that we had a greatly improved interface, and which was very important. It's a user-friendly and gives us a readily available display of information, which is fundamental for the process of choosing, as I said, the right tool. There is a large and I believe growing curated inventory of genes that recognize as pathogenic and involved in major human genetic conditions that we can use, and this is a big part of the work that we used to do before. This system allows viewing gene maps and expected coverage, so we know what to expect from experiments, which is a very important information we didn't used to have. The genes are organized in different areas of pathology, and this provides essential information about each gene and associated phenotypes, and it's important to have them classified hierarchically into categories already. The repository genes can be arranged in any combination, and this is very important. So they can be analyzed in panels of a wide range of sizes. The system provides high flexibility both in terms of content and possible integration, and especially the possibility of building comprehensive targeted panel, which is our goal. And the wet analysis procedures are absolutely optimized. So the key element, the gene prioritization, is a work that we find done already. So this is a big part of what we used to do, and it's a fundamental help. The interface is friendly, but again, it's fully informative, and that's important. The genes can be assembled with high flexibility into panels of different composition and different sizes. And the chemistry of the new system is definitely a big improvement in the workflow. So at the end of the story, I think we have we hit the target. We are not chasing a moving target, or at least it's not as moving as it used to be before. We have the possibility of assembling comprehensive disease panels with great flexibility, both in content and timing, because we can decide of modifying our panel whenever we want and however we decide. And with this, I thank you for your attention, and I'm happy to answer questions. Thank you very much.
Thank you very much, Dr. Murgia, for that informative presentation. Next, Dr. Hernandez-Guzman will provide a live demonstration of designing a custom gene panel using Ion AmpliSeq Designer. But before I turn it over to him, I would like to remind our audience that we have a couple of poll questions that we would like for them to answer. And so if you could take just a quick moment to do that, and then we will move on to Dr. Hernandez-Guzman. All right, thank you. And now I would like to turn it over to Dr. Hernandez-Guzman for his presentation.
Great, Q. Can you please just kindly confirm that you can see my screen? Okay, I think hopefully you can see my screen okay. So what I'm going to focus today is really getting a little bit more hands-on with the whole process of creating a panel. And as Dr. Murgia mentioned, we are using a website called ampliseq.com. And when you first visit the site, this is what you'll see. If you're currently a user, then you'll be able to just sign in. If you're not a current user, then you just click on the register new account. And it only takes a couple minutes to register an account. You just have to provide your information, and then you'll be able to access the website. Once you've accessed the website, what you can see is the website is organized in 3 different sections. It's organized in ready-to-use panels. These are typically fixed panels that come in small reaction packs, and they're organized between DNA and RNA. And these are really panels that have been in our inventory for a while, and people have been using them fairly successfully. On the right side of the screen is what we like to call the made-to-order panels. And Dr. Murgia's, in her experience, she used some of these panels in her earlier studies, which she was able to do quite successfully. And this is really a very flexible tool that allows you to go not only from a gene level to a region level, and it offers you different amplicon sizes for different applications. It's extremely flexible. But what you do see is that you're tied to a made-to-order process, which basically means that whenever you create a design, the design needs to be actually computed in the back end, and that can take some time, and then it has to be manufactured from scratch, so that can also take some time. So there's some advantages and disadvantages to use the made-to-order versus our new product that we have today, which is really much more focused as an on-demand panel, meaning you can create this design immediately. The content is available in inventory. It can be pulled together rather quickly. And also you have other key benefits, such as you have small reaction packs. The performance has been optimized, and you can actually get a preview of the performance that you could expect in the lab. This is something that you don't typically get with a made-to-order panel. With the on-demand panels, you actually get a preview of how likely it is the panel to perform. The on-demand panels are focused on human and the human genome, and it's really more related to human disease research analysis. There's two entry points that you'll be able to get access to. Is if you already have a list of targets that you're interested in, then you can just proceed to the list. Let me just kind of preview that for a second here. You can start typing in your gene names, or you can choose a file where you can just input a series of gene lists in a batch form. However, what I will focus in today's demonstration is going to be really using the disease research areas, because we find that this is basically content that has been organized by gene associations to different diseases that have been studied by various public databases. So when I click on the disease research areas here, what you'll find is a top organizational model here, where everything is organized basically in alphabetical order, and you have many different sort of high-level organizational diseases. So for example, if you're interested in cardiovascular diseases, you can just click on cardiovascular. It goes down into, maybe let's say I'm interested in heart diseases, I come down to that. And maybe I can still, let's say I'm interested in heart failure, I can go down to that level, and then select sort of subcategories within those areas. I can always kind of go back to the initial sort of high-level organizational, and then at this point, let's say that I am interested in some of the heart diseases, and maybe I'm interested in, let's say, the heart failure that we talked about, and maybe I can have also diseases that are related to heart valve conditions, so I can select those as well. Right now what the tool is doing is basically storing that selection at this level. You will notice that there is actually a limit as to how many genes you can actually expect, and there is a maximum of 800 genes that we can accept, although when you go ahead and order, we can actually create such a large panel up to 300 genes, which is really, really large at this point. But when you start in your selection exercise, this is the way you would go ahead and do it. So at this point, let's say that I'm ready to create my first panel. So this is webinar, and then I said this is panel number 1. What you can do is just click the proceed button, and what is going to happen now is the tool is going to assess the content of those genes, and then it's going to go ahead and do a search of what is currently available on demand and separate the content that is not available on demand for you. The content that is available on demand is the content that is being available in inventory, and also there's performance metrics associated to it. So for example, let's say that I'm interested in the RAF gene right here. What you'll notice here at the bottom is there's a track called expected coverage. And in the expected coverage track, what I can do is I can start zooming in to really get a better understanding of my region. So for example, let's say I have a variant in this particular exon. I can zoom in there and try to figure out, okay, what is my expected coverage level in terms of number of reads or the depth, and I can see, okay, well, everything is covered nicely. I can proceed with my design. If I click on a different gene, then the IGV viewer that has been integrated into our tool updates automatically. There's also a gene information table that's called also a meta table right here. and you can also have other useful links to other publicly available websites, as well as an interesting point here of what is this gene associated with as far as other diseases that might be interesting for you to study. So just because I started with a particular set of focus, let's say in the cardiovascular disease space, I can notice that now this gene is also involved in mental disorders. So these associations are actually more of a matrix type of associations. They're not just necessarily linear based. So a single gene can be associated with multiple diseases, which is a very interesting thing to learn. So at this point, users, if you do see a particular panel that you're happy with, this is a 61 gene panel. But let's say that you're missing a gene. Let's say I want to include, for whatever reason, I want to include a tumor suppressor here, just as an example. In this case, it tells you, it's very basically smart enough to tell you that that tumor suppressor is already included in your list. So then life is okay. Let's say you want to include a different one. Let's say I include the infamous BRCA1 gene. What it will do is actually add it to my list, and it will just label it here with a little dot to just say, hey, I added this after my initial selection. So it's easy for somebody to say, okay, this is my first selection that I've created, and then I have my additional content that I created. You will notice that there is a set of genes that are not available in our inventory at this time. And as Dr. Murgia mentioned, our inventory is growing, so you could expect regular updates throughout the year where you have additional content being added. So maybe today I may have, for example, the ELN gene not available in inventory, but maybe in a few months from now, the same gene may now be part of our available on-demand inventory. So I would encourage you, if you start creating designs, whenever we have a new release of the software, you may want to look back and see, oh, okay, some of that content now I can get available on demand. Now, when you have the content that is available on demand, then it's very easy for you to just proceed, because this particular design that has this experimental data information available for you is basically ready to order. There's really no creation of the design. Even though the download results function is disabled at this time, the design is ready to go. So you can just click the order button, and in here you will have the option for doing a manual workflow, or if you have an Ion Chef instrument, you can also select that, and that updates the library kit that you want. This is actually optional. You don't have to include it in your order. If you do, it gets processed with the order. And once you click to the order button here, what this is going to do is going to move you to the pre-card option here, where you then have other options. Let's say you're interested in adding additional, let's say for the S5, you might want to add some chips you want to your order, and then you can proceed to the cart. So, but let's go back here, because one of the very important questions that often happens is, okay, you have this nice organizational, and actually if I look at it as a table view, you can see that, for example, my elastin gene is quite important for because this is actually based on association score. This is quite important for my particular gene. So let's say that I do want to include that gene as part of my overall design, and maybe another gene over here, just general transcription factor here. I can go back to my grid, and you'll notice that these genes are actually being selected here. What happens is that what we've put together is, because we knew that there would be certain genes that would be important for you that may not be necessarily be on our inventory right off the bat, you will see that there is an option here for creating what we like to call a spiking panel. And what the spiking panel is going to do is it's going to leverage our made-to-order process for creating this design. And when you're ready to order this particular design, it will be ordered in a concentration that is compatible with spike-in. And what that means is that the original panel, when you go back to your lab and you have your two-pool panel, you'll be able to spike in this panel to add effectively coverage for this additional two genes. From the bioinformatics side, all the bed files will be updated. Everything will be ready to go when you run a spiking panel. So from an analysis standpoint, you don't have to worry about it either. But this is a very nice way for us to be able to offer as much flexible content as we can without making any sacrifices as far as having to wait till the content is available in inventory. So it's actually quite an exciting experience here. So once you click the order button, then this design will be locked, which means that all the files get created for analysis. If you do want to go back and make modifications to that initial locked design, we have a very nice feature here called the clone panel. What this is going to do is basically going to copy everything that you have in your initial design and basically open it up again so that you can edit that particular design. So at this point, I think that I've given a fairly nice demonstration here of how easy it is to create these on-demand panels. The last bit that I wanted to call out is the videos that we have available. There's multiple videos. One is right here for just getting started. There's videos that is more specific to working with the disease research area content. There's also a short video also for the gene list. We've actually enhanced our help. If you go back into the frequently asked questions, you can see that there is a series of questions specifically targeting the on-demand product, so that you can go back and maybe if you have some questions that you want to see if there's an answer for, then you can go quickly there and try to find more of a self-serve type of experience. So with that, I think I'd like to open it up for questions at this time. Thank you very much.
Thank you very much, Dr. Hernandez-Guzman. And thank you both for that informative presentation. Before we get started on the question and answer session, I would like to just remind our audience how to submit questions. You can submit your questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Just a reminder that any questions we do not have time for today will be answered via email. And is going to be for Dr. Hernandez-Guzman. After you customize a panel, what are the typical reaction sizes you will get?
Yes, so if you're working with the on-demand product, if you're using a manual workflow, you basically get 24 reactions. And the price, it would be according to those number of reactions, so it would be fairly accessible. If you are using an Ion Chef type of protocol, then you will get an A reaction size offering. These are the reaction sizes that in the future we're looking to expand so that people have a little bit more choice. But for the on-demand, it's basically two options for reaction sizes, depending on whether you're using a manual workflow or an automated workflow.
Okay, thank you. Our next question is for Alessandra. What are the top features of the Ion Torrent systems?
Well, I guess I've insisted on that in my presentation, but I guess generally speaking, the Ion Torrent system is tailored to the need of a single lab and is the answer to whoever has the need of doing next generation sequencing in a let's say clinically related in a clinical research situation where the data need to be translatable and actionable immediately. The next generation sequencing of the Ion Torrent is innovative and is more sustainable, in my opinion, than others. The characteristics of the new systems are finally those that will allow to have flexibility in choosing targeted panels, which is what we were sort of expecting, because To work with panel very often is to work with something that we keep chasing with a situation that keeps moving. So we need to be flexible, and we need to be able to adapt this flexibility to our experiment. If I want to expand my panel because there's a new gene and I find that a new gene in the repository, I can just add it. And I can just add it at different times, which gives me a better option, which gives me what I really need instead of a closed system. To work with panels is, in general, working with a situation where we have this confidence of being able to cover a certain clinical condition, of being able to have the feeling that a negative result is actually negative, and we are not, hopefully not losing information. So I think these are the key factors: flexibility, confidence of having the possibility of covering extensively a clinical phenotype, having the possibility of having a comprehensive coverage. I guess these are the main features.
Great, thank you. Our next question is for Dr. Hernandez-Guzman. How many genes can I add to a spike-in panel?
Oh, yes, that is actually a great question, and thank you for bringing this up. So the maximum number of genes is actually, the metric that we use is number of amplicons that can be added to a spiking panel or a spiking reaction. In this case, it's actually 123 amplicons per pool. So that's a total of 246 amplicons for the two pools. And what that really translates to is approximately around 10 to 12 genes. It's kind of hard to give a hard metric because obviously not all genes are of the same size, so some require more amplicons than others. So really the metric here to look for is as far as number of amplicons that you can have. But on average, it's anywhere between 10 to 12 genes could be added as a spiking reaction.
Great, thank you. The next question goes to Alessandra. What are some factors to consider when choosing targeted panels other than what you already shared?
Well, I guess one of the most important things now is to address the problem of depth coverage for two particular conditions, and one is somatic mosaicism, and one is probably gene dosage. We know that gene dosage has always been a problem, so we cannot address gene dosage with sequencing, but next generation sequencing has the capacity of being reading with enough depth that we are getting there. Now, what we didn't expect, or we didn't expect at this extension, is that we had to deal with somatic mosaicism, and it's becoming more and more clear that even with Mendelian disorder, this is a problem we have to address. So we have to have the feeling of having the possibility of detecting variants with a high confidence, but we also want to be able to detect variants that might have a different representation than the classical allelic one. So I guess for this reason, for these two problems, for a problem of depth of coverage in these two conditions, there isn't any other option than going with panels that can give us an extremely high coverage and an extremely extensive coverage of the sequences we want to analyze.
Okay, thank you. Our next question goes to Dr. Hernandez-Guzman. Is there any information provided to make sure that the panel I'm designing will have enough coverage?
Yes, so this goes back to the view that I showcased during the demonstration, which is really looking at the IGV viewer. There is that track called expected coverage, and what that track means is basically that for those genes that have that information, we ran them in the lab and we observed a particular sort of performance metric, and we reported that in that particular track. So when you're looking at making sure that you have enough coverage, it's not only about sort of making sure that the exon region is covered by the amplicon, but it's also understanding how well that is covered. So the actual sort of overall coverage metric, which is not to be confused between in silico coverage versus expected coverage. Those two factors are a little bit, have a tendency to get confused. But we report both. We actually report our in silico coverage, which is how well were we able to put our primers in such a way that the amplicon covers the whole exonic region or the CDS regions. And then the expected coverage then is, okay, what is it that we observed in the lab? So we report both, and those can be accessible from the IGV viewer as I showcased in the demo.
Thank you so much. That's all the time we have for questions today. I would like to thank our sponsor, Thermo Fisher Scientific Ion Torrent, for making it possible to bring this presentation to you. Today's webcast will be available for on-demand viewing through January of 2018. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. We'll see you next time. Goodbye.